My work focuses on how endothelial cells within a blood vessel responds to different levels and type of shear stresses that the flow imposes on the cells. The blood flow in the arteries produces a frictional force called Paul shear stress on the vessel wall lined with endothelial cells. The endothelial cells are shown to express pro-inflammatory genes and hypermeability in response to disturbed flow with low shear stress, while they show anti-inflammatory phenotypes in response to laminar shear stress. Yet, it is still unknown how the endothelial cells distinguish the different flow shear stresses. Our hypothesis is that the fluid shear stress is transferred to the force sensor attached at the cell-cell interface and at the cell extracellular matrix interface through direct force transmission via cytoskeletal body of the cell known as cytoskeleton. My goal here is to test this hypothesis by measuring mechanical responses on the extracellular matrix and cell-cell adhesion sites while applying different types of flow to a monolayer of endothelial cells. To measure the forces of the cells and the flow, I have developed an in vitro flow channel with soft gel surface cover slip coated with fluorescent microsphere to track the forces due to flow. The flow channel is then hooked to a flow system to provide a healthy environment to the cells for live cell imaging. I grow the endothelial cells into a monolayer on the soft gel in a flow chamber, then start a physiologically relevant flow to the chamber under the spin-disk confocal microscope. I image the cells and the beads on the gel during the flow and image the beads again after the experiment to record the relaxed state of the gel. Using the software developed in the lab, we analyze the image for the distribution of displacement and traction forces. The current result is one from many sets of experiment which shows the membrane of the cell and how the force is transmitted on the soft gel substrate over time after the flow. This example shows that within 20 minutes after a laminar flow, cells show a reduction in overall deformation and the force to the substrate. I'm in the process of refining the flow chamber so that I can image the cells in a more robust manner. This force measuring framework in response to the different type of flow will be coupled to the analysis of cell-cell and cell extracellular matrix adhesions through the evidence of potential force transfer and the associated molecular pathways. And this might be helpful in improving the therapeutic applications by targeting those pathways.